So I'm very pleased to introduce our student invited speaker today. Rafael Irizarry is, we have a few titles. I will try to get them right. Chair of the Department of Data Sciences, Professor of Biostatistics and Computational Biology at Dana-Farber, and Professor of Biostatistics at Harvard. Professor Irizarry has been very influential in lots of fields of computational biology, RNA-seq, microarrays, sort of every new field of computational biology that pops up. He has some great contributions. So I'm really looking forward to this talk.
Thank you. Thanks for the invitation. Thanks to the students for inviting me and for picking this talk, which I haven't given in a while. Start right away. So you'll in a second, you'll hear about what this music is. It's related to the talk. So what I'm gonna give you a summary of 3 things I've worked on throughout my career, and one of the things I'm going to try to do is focus on the data science aspects. I know that that's like a term that's being used now, and it's maybe confusing some of us, and there's different ways different people use it. So what I'm going to do is I'm going to focus on the aspects of all these research topics that aren't necessarily taught to you in grad school. And then that, in a way, that's a little bit of how I think of the distinction between data science and statistics. Is a lot of what we have to do to get to the point of analyzing the data. And if you want to hear more, learn more about my thoughts on that, I actually wrote a paper or opinion or whatever you want to call it, that you can find in the Harvard Data Science Review. So I won't get into that topic, but I will focus on the talk about on that aspect of my research rather than the technical stuff that gets published in journals. So the 3 topics I'll talk about are music, sound signals, high throughput gene expression, and then I'll end with a short summary of our work related to estimating mortality in Puerto Rico after Hurricane Maria. So I'll start with music signals. So again, I'm going to focus on the what I would call more data science aspects of my research, not on the technical ones that I did write up at some point in my career. So we won't be seeing any of that. Instead, I'll be focusing on practical aspects, wrangling the data aspects, and getting the work out there. That kind of stuff. All right, so I'll start to motivate the first part of the talk. I'll start with the explanation of what that music you were hearing at the beginning, what that was. So that was a soundtrack from the movie Farinelli. So Farinelli was a very, very famous singer from the 19th century, and he was a castrati. He was like, on his time, he was a rock star, very, very famous. They made a movie about him. As far as we know, there's no more castratis to play his part. So to get the sound of the singer, they had to create a sound that didn't exist. And to do this, they recorded the whole score. A female soprano recorded it, and a male, I believe a male alto recorded the score. And now with those 2, they merged it in a way that made it sound like what they thought, or actually they had some recordings. There's some early recordings of it. So they try to imitate, they try to mimic that sound by combining the 2. But then to understand how this relates to data, I'll tell you a little bit about music theory and sound signals theory. So one of the things to start to motivate that, to do that, what I just described, if you were able to get the data of those signals, the male and the female voices, if you add those 2 signals, like and then you play it, that's going to sound like 2 people singing together. So you can't just do that. But there's a transformation you can do and then add them that will work. And that's where I'm going to try to get you to understand today, just a little bit. So when I was this was my PhD thesis work. So when the way I got involved in this is because my advisor, David Brillinger, when I asked him to give me some of his papers to see if I wanted to work with him, he said, no, I'm not going to give you any of my papers. You tell me what you like. And the only thing I could say was music. And he said, okay, we're going to work on that. So he went and found these people in Berkeley that were working on sound signals. And they wanted to work with a statistician. They wanted to help them decompose the sounds in a way that they manipulate them to make new music. So one of the first things they showed me was this. This is a sound that they wanted to break into parts. I don't know how loud that was. I hope you heard it. Let me see if this has volume. Yes. Get louder. Give me a second here. All right, let's hear that again. So you can hear it goes, it has a wavering of the pitch. It goes, and also you can't really hear it that well on these speakers. Hopefully some of the others will come out stronger, but there's a lot of wind sound. So they wanted to decompose that. They wanted to separate out the more flute-like sound from the wind sound, and then try to do. They were more artists than scientists. They were both, but they were very artistic. They wanted to make new sounds and compose with these kinds of signals. So we started helping them, and to help you understand how we got to helping them, the first part of it was quite difficult, was to get data. So to understand what that even means, I'll explain a little. Oh, you wanted me to use this?
No, I thought I didn't restart the recording.
Okay, it was, right?
But I did
So I'm just going to try to give you enough to understand how this could possibly be related to statistics. So the first thing to know is that sounds they're signals. So you can think of it as a continuous function. I don't know how many of you have ever seen that object up there on the right. That's a cassette. And that's what we used to use to listen to music. That we record music on those things. And what was recorded on those things was actually a version of that function, continuous function. But then later in my life came a new technology, which is you might have never seen either. It's a CD. And the CD did not store the function VT. Instead, it stored discrete, like sampled versions of VT. They sampled it at a 44,100 times per second. So it got very close to being exactly like VT, but not exactly the same. So that's where we can get data from. So it wasn't easy at that time. Now it's actually quite easy, but getting stuff from that CD to data. This is a plot that I made in 1995. I made an S+. You might not even know what that is, but before R, there was something called S+. And there you have it. That's a violin sound. It's like maybe 1/100th of a second of it. So now I have data, and we can start trying to help these collaborators. Okay, so let's keep going and try to understand a little bit more. So the second thing you should know about musical sound signals is that when they're periodic and the frequency is between 20 cycles per second and 20,000 cycles per second, you can hear a pitch. You can hear what you consider to be a musical sound. So that is something we can use to do what they wanted to do, which was to separate out the flute sounding part from the not flute sounding part. They actually wanted that part to play around. So if that is something we know, we can start using this information to try to do that. So this is plot that I'm showing you here is something called a periodogram. And what it is, it's basically a regression. You take your data and you regress it on sine waves. And if it is moving, if it does have a periodic behavior, then then the frequencies, the sine waves with the frequencies that match that will be high and the rest will be low. So that's what that's showing you. So this is something that was first observed in 1885. My thesis cites an 1885 paper. And that was done not with a computer, obviously. So this guy, von Helmholtz, had like little glasses that would vibrate only at a specific frequency. And when these sounds came along, then by seeing which ones were vibrating, he could tell that there was some kind of periodic thing going on. So you see a bunch of peaks. That's because these sound signals aren't sinusoids. They're much more complicated than that. So when you have this is a famous theorem in mathematics. If you have something periodic, and you convert it to the spectral domain, you regress it on sine waves, is another way to say that. You will see peaks not only at the main frequency that the sound is coming at, but at its harmonics, at its multiples. That's actually a mathematical result, right? And you'll see the formula soon. Now, so it's not a sine wave. A sine, if you listen to a sine wave, it sounds like a beep on a computer. Very boring. It will probably annoy you after 5 seconds. So different instruments will have different shapes, and that's kind of what determines the pitch. But there's something else that's important. When you hear a sound, it doesn't go on forever. So it really can't be completely periodic. And it also has different other things going on, like, and I'll say that later, but then what's happening is that these shapes are actually changing in time. So here, if I show you this picture, it's actually kind of hard to tell them apart. That's a periodogram for a trumpet, and that's for a clarinet. Clarinets have kind of square waves, and a square wave has higher peaks on the odd frequencies and the even harmonics, and you can kind of see that 3 is bigger than 2 and 5 is bigger than 4. It's little factoids. But when we do a periodogram across time, this is a 3D version of the previous one where I show you hundreds of periodograms next to each other with color. Now you can kind of see that different instruments are actually quite different. So one thing you see is that there's instruments that can sustain the sound consistently, especially the wind instruments like the oboe. So you see that it's kind of goes all the way. None of the harmonics stop. The violin is a little bit like that, but it's a little bit harder to sustain. So you see that some of the higher harmonics start dying off. And then you have an instrument like the guitar where where it's not sustained at all. And you just pluck and you hear it and it goes away. And you can see very clearly that the higher harmonics go away. But the main point here is that there's definitely some signal that we could model with harmonic models, with sine waves. And we can describe the different instruments by how strong those signals are and how long they last, how strong they are differently across the different harmonics, all these things you can do. Then here at the bottom, I have what are called non-harmonic instruments. Although some of them, a marimba is a little harmonic, you see a stripe there, but the gong is definitely not. You don't see those nice harmonics because it's really not periodic. Okay, so that's a 10-minute explanation of sound signal theory. Now that we've seen the data, we've explored it, we can actually try to model it. So I'm going to write it like that. There's the data Y, and then there's a signal, a harmonic signal F, and then there's the rest. Whenever I call it noise, the musicians would get really annoyed because that's what they wanted. They didn't want, it was the more interesting thing to them. They wanted, they made me call them residuals, which is good enough. So that's what they were interested in getting epsilon and hearing epsilon and playing around with epsilon. But for my thesis, I had to prove theorems and whatever, so I actually focus on the whole thing and prove that the parameters converge, all that stuff they make us do as statistics students. But this is the model basically that we came up with. It's not that complicated, but basically it's a sum of sine waves. This is a harmonic model, and it's changing through time. Right? So that there's Well, yeah, so it's changing the time because this only applies for a small window of length h. So as you start moving t0, then you start getting a different set of parameters. So we got that. We then recorded. We had to record some instruments to start figuring out how to fit the model. They had the collaborators helped us with that. So to get the data, we actually had to have someone come in with an oboe and play it on a microphone and record it, and then they would give me this file that then somehow got made into a file I could read into S+. So this is actually fitted model results. That's the signal up there, and that's the F. Not really very useful to look at that. These are the residuals. This is the estimated lambda, and you can see it's going up and down. That's because this oboe player was playing vibrato. They were making the pitch go up and down. It's only deviating a little bit. It's not changing the note. It's just making it go a little bit up and down. And then these are the row Ks, the size of the harmonics, and they're also moving around. So let me just play that for you. This is what that sounds like. Whoops. It's an adventure. That one, that's the sound. Okay, so that's the sound. Now, what I'm going to show you now, this, looking at this wasn't, if I wanted to know if the model was fitting or working, this doesn't really help that much. It does help a little bit that I kind of expect that to happen with an oboe, where at the beginning there's more residuals and later stronger residuals. That's because there's a burst of air at the beginning. They're like, they put that you can actually hear that if you listen very closely. So you kind of know it's right, but the better way to do it is to listen to it. So this is residual analysis by ear. So this is the clarinet fit. This is the original, the fit. They'll sound almost identical to you. But what's much more interesting is the residuals. Sounds like air going through a tube, right? That's what the clarinet is. And then the violin, you can kind of guess what that's going to sound like. Sounds like screeching metal. And the guitar is going to sound like a plucking metal. Oh, you can't hear that one, huh? All right, sorry. The sound system doesn't work well enough to hear that. Can you hear it? I could hear it here, but it sounds like pluck. So that's telling me it's working. So this is the final version. I had to play around with several things to get to here, because sometimes you would hear this signal in the residuals and you were underfitting. Sometimes the residuals would sound too much like white noise, like not having any structure to it. So then you were overfitting. Okay, and then the last thing I wanted to show you, this is very cool, I thought. This is a so let me explain what this is. So musicians say that if you listen very, very closely to a oboe, you can hear a hidden soprano. You can hear the voice of a soprano singing the note at double the pitch. My ear isn't good enough to hear it, but that's what they said. So what I did was, I thought, well, if that's true, that means that if I play an oboe and I shake the even harmonics, I should hear the female singer that's supposedly hidden in the oboe. So now that I have the data, now that I have the model fit and I have the parameters, I can actually do that. I can make the row Ks go, I can manipulate them to go up and down, then convert it back to the fitted signal, and then convert it back to sound and listen to it. Well, here it goes. Let's see if you can hear it. So what you're going to hear is it's going to start as an oboe, then I'm going to start shaking the even harmonics. And if the sound system works good enough, we'll hear it. Put this all the way up. Damn it. Requires some dexterity. All right, I'm gonna try one more time and give up. All right, here we go. I don't know how, I could hear that. I don't know how many of you could hear, but then the first time I heard that, it freaked me out a little bit. All right, so definitely sounds like a woman singing. Okay, so then to come back to the story of the first thing they asked me to do, I won't play the sounds for these ones. You can show them to me later if you want. But this is that flute at the beginning, and they wanted to hear the residuals. So this is where one of the harder statistical challenges came in, is because as I'm fitting the model, once I pick a window, I feel comfortable with it. I just use it the whole way. But with this one, because the sound was stable and then changed, you can see it in the fit up there. It needed to, the size of the window needed to change. If I just use a single window size, you can see in the residuals that the residuals grow over here. And if you listen to it, actually, I'll try to play that one. You'll hear in the residuals that you underfit the model. Because I can hear the flute in the second part. That you heard like wind, and then it went wheel, wheel, wheel. That's because I underfit, and that you can see it. You can actually see that visually too. Then if I do it, if I say, well, let me make the window size smaller, then I overfit. And the way you can see that is because the residuals no longer sound like wind; they just sound like white noise. So one of my thesis papers was a method that would automatically. I couldn't be picking the window size by hand because these were, many windows. So I wrote an algorithm that would change the size of the window depending on, I can't remember. It was like the title of the paper is information criteria approach. So now you can see that it's kind of a better balance. And these are the window sizes that are picked over here on the top, and you can see that when the flute is shaking, then the window size gets smaller. And now the wind sound that they wanted and they were happy with, you can hear here. You still hear a little bit, but that was maybe I put the wrong sound, but the final product just sounded like wind, which was exciting to them. All right, so that was my project with music. And now I'll give you the second example related to high throughput genomics. This is a gene expression example. So I like to motivate this part of the talk when I'm talking to students with this guy, Anton van Leeuwenhoek. He discovered a bunch of different things. There's some examples of many of the things that he discovered, and you can see the common theme is they're all very, very small things that we can't see. So you think, oh, well, this guy probably invented the microscope. He didn't invent the microscope. He just made it a little bit better. So the microscope was around and people were looking through it and looking at water from a puddle and couldn't see much. But this guy made it better, and then he all of a sudden could see things that others couldn't. So I like to use that as an analogy to what we're doing, a lot of what we're doing in genomics, which is we're taking a measurement that has something in it that this might be an interesting discovery, but it's very hard to see, and you got to put it in focus to really see it. So, and in this case, it's even more extreme because the data, for example, with sequencing comes in as a bunch of letters, and that's the data file for a sequencing experiment. So there's, I think, 1 billion lines like that. The biologists can't do anything with that. So computational people and statisticians convert that into something that they can see and interpret. So this is an example for a project I worked on where we use sequencing to try to find, this was a case where we're trying to find regions of the genome that were methylated differently across tissues. And using smoothing techniques, we were able to find them, like starting with those sequences and going all the way down to being able to show the biologist something like that figure where they can say, oh yeah, I can see the differences. So that's how I think of what we do. So I'll give you an example of some of my earlier work where we put the data into focus. So this is data from 5 different tissues. This is real data. None of what I'm showing you is simulations or cartoon data. It's real. You can download these data sets from Geo. So there are 5 different tissues, and for each one I have several individuals. So you can see there's 5 colors, one for each tissue, and there's several points for each one because there's several individuals. So imagine that we didn't know that what makes tissues different. So I had a picture earlier where I showed a bunch of different stages of development. The point of that plot was to show you different things that have the same genome but look very, very different. And the way that, and also different tissues are same genome. So your colon and endometrium, same genome, different tissues. One of the ways we think that happens is that different genes get expressed. So even though those 5 tissues, if you look at the DNA, they're all exactly the same, they're very different tissues. And the way that happens is that different genes emit different signals, different genes get expressed, and that eventually leads to the tissues being different. So suppose we didn't know that, and we have expression measurements from those tissues, and you want to see how are these tissues different? Does it have something to do with gene expression? So if I download the raw data from these experiments and I process it with the algorithm that was originally designed for this particular technology, this is a PCA plot that I get for these vectors. Because remember, each one of these, it's measuring expression for all genes. So each one of these points represents 20,000 dimensional point. I'm reducing it down to 2, and there's about 100 of them. So I make a PCA plot, which then means that the distance between those 2 points is a very rough approximation of the actual distance between the original vectors. So if that's, so if I look at this picture, I don't conclude that different cell types have different profiles of expression, because they're kind of all over the place. If you look down here, so the bottom green point, so this green point is closer to this pink point than it is to this green point. And there's many examples I can point out like that. So we see a plot like this, and we'll ask ourselves, oh, this data looks kind of out of focus. Why is that happening? So when we had that, we asked for the raw data and started to look at it to see if maybe there was something going on that we could fix. So this is a plot of the raw data. Now I'm showing you, instead of 100 different samples, different samples from different cell types, I'm showing you a plot from just 2. So I'm showing you a scatter plot, but it's been transformed so that the log ratio is on the y-axis and the average of the 2 measurements is on the x-axis. So you have highly expressed genes over here on the left, low expressed genes over on the right, different ones on the top and the bottom, right? Those are log ratio big, very high or very negative, means they're different when you compare them. The gene expression for that gene is very different in these 2 samples. So now I'll reveal that this data set is actually a data set that was created exclusively for us statisticians to figure out if the technology is working. It's the same RNA separated at random and put on the measuring device. So in theory, all those points should be on 0, because it's the same, well, maybe some noise, but there shouldn't be any difference. With the exception of the 16, is it 16, maybe less, 12, something like that, orange points, which are the lab added them in a way that they should be different. So the orange points should be different, the black points should be at 0. And that's not what we're seeing. So when we first encountered this, we started asking, this doesn't look like it works. It's not, there's too much noise. And we got a lot of explanations about it. One of them was, this is how biology is. And another one was that this technology is going nowhere. It doesn't work. But we got our hands on the manual and we looked at how actually this ended up getting to be a number. And we found out that there was actually even raw data that got processed before getting here. And we found something that might explain this. So here's what it was. So to get to that plot, they take two measurements for each point. They take two measurements for each dimension of each point. They measure what they think is the gene expression intensity, a measurement that's supposed to tell you how much gene expression there is, but they know that there's probably background noise. So they took another measurement to measure the background noise. So that's what alpha, so they're thinking that you see the two measurements they take for each one of those points, each dimension of those points, y1 and y2. The first one is measuring what you want to know, which is beta, that's gene expression level. And then there's an alpha, which is the background noise, which you don't want. But if you just measure y1, you won't be able to know which is alpha and which is beta. It's not identifiable. And they also knew that different measuring units would have different alphas. So that would make things kind of move around in a way that they couldn't predict. So they actually try to measure alpha and then subtract it to get beta. That was the idea. So we asked our collaborators to run an experiment with no beta. So this just putting random stuff on the measuring unit. And this is what the plot looked like. So this is the y2 and this is the y1 in the log scale. And you can see that it does look like, so in this experiment, there's no beta, so they should be identical, but they should both be alpha. They do look somewhat identical, but there's a lot of noise. It's expected that there'll be noise, but if you now look at what they're doing, and then you realize that when people are comparing these things, they're looking at the ratios and which you then usually take the log ratio to make it symmetric. If that's the case, if that's what they're doing, and from the data we see that the correlation between the alphas is about 0.7 in the log scale. So by the way, this is the better model, as I'm saying here, is that instead of alpha in both y1, 2, there's an alpha 1 and an alpha 2, which are correlated, but they're not the same. So this subtle statistical insight now tells us, right? So that's the model. It's a little bit different. Now the alpha is random, it's correlated, but not the same. That tells us that you can now do, like using Taylor's theorem or whatever, you can show that the variance of the log of that correction is going to have a variance that increases with the square of the reciprocal of beta, which is kind of what we see in this plot. You can see that 1 over beta squared, you can almost see it there. Like this is related to beta, and the variance seems to be related, it seems to follow that pattern. So it does look like the statistics tells us why that's happening. So it's not that biology is like that, and it's not that the technology doesn't work. It's just that they made a bad decision when correcting this measurement. So what we did is we there's many ways you can do this, but now you try to estimate beta given y1 and y2, and statisticians can have a field day with this question. How do you model it? Are you Bayesian? Are you empirical Bayesian? You want whatever it is. You can come up with many different solutions, but now you focus not on their approach, which focus exclusively on reducing the bias. Now you can come up with an approach that reduces both variance and bias. So you don't want that thing to blow up. So you come up with some other approach. We had many ideas. One of them we implemented, and we were able to take this data, same data, go back one step, take the raw data, put it through a statistical algorithm that reduced variance as well as bias, and then it turned into this. So that is now you can actually see the orange separating from the black. So that was published in 2003 and in a statistics journal. So now that was just that experiment, but how about the original thing that I showed you? Would we be able to make the discovery if we use now this other algorithm? Well, now when you take this out of focus data with the original approach, you apply the new one, now it turns into this. And now you can actually make the discovery that different tissues have different gene expression patterns. So that's my second example. And one of the things I like to share about this is that paper has been cited quite a bit. And my explanation for the main reason is that we made software for it. This is an old screenshot. I don't think it's number 16 anymore, but because that technology is now not used anymore. But we made a R package, made it free, open source, and then a lot of biologists that had this data and didn't like the results they were getting started using our method instead. So then they get the software, and when they're going to say how they process the data, they just have to cite us. That's how we got those citations. Okay, so that's my second example. And then my third and last example is a project where we were trying to estimate mortality rates after Hurricane Maria. So I'll give you a little bit of a background on why this was even a question. So this is in 2017. This is an aerial picture of the hurricane. That's Puerto Rico there in the middle. You can see how big the hurricane was, Maria. That's some picture of what it did. This is the department where I studied math. It's gone. It's now gone. At least the structure. They still have a math department. And the way that we started working on this, the motivation, was because I have family and friends there. A collaborator of mine had a colleague that worked in the field in Puerto Rico, and from both sides we were hearing that this was really bad. The hospitals are shut down. There's the ambulance, 911 isn't working. It looked like it was very bad. And this is in October 3rd, I believe. And it was about 2 weeks after. And the governor is claiming that only 16 people had died as a result of the hurricane. And that just didn't match none of what we were hearing anecdotally. So a colleague came to talk to me about possibly trying to figure out what the actual count was, Carolyn Bucky. And I said I was more than willing to help. So our first attempt was to get data from the demography department. Puerto Rico has a stable demographic department that records every death. And from there, you should be able to see if there was some kind of uptick. But that was not happening. They weren't sharing data. So she works in problems like this, and what she thought of doing was trying to estimate it by going to the field and having a poll, doing a poll. So you go ask people, has anybody in your family died? And then from there, try to estimate. So they were planning that. You got to get IRB approval, so it takes a long time. It's a complicated thing to do. In the meantime, we're starting to see reports already anecdotally saying there's something more than, it looks like it's more than 16. So this is the first one I saw, November 8th. And this is New York Times. And then what they did is they're noticing an uptick in funeral homes. Here's another one. This is a survey of funeral homes, same kind of conclusion. Then on November 21, a bioarchive paper comes out, and they seem to have data from the demographic registry. So again, we asked the demographic registry if they can share the data, but this is now saying using data from the demographic registry, which as far as we know was not public. So we're not sure, we still to this day don't know exactly how they got this data. But from there, they can compare October, September of 2017 to previous years and already see there's 1,000 more deaths than expected. You can get in that data, that paper was published, you could scrape the data from the paper and see what they were seeing. They had, they basically are doing this. They have, the black line is 6 months, is the average of 6 years previous to the hurricane. And then the blue points is 2017. And you can see how obviously September and October right after the hurricane are way more deaths than expected. Then shortly after that, the New York Times also got their hands on the data. The reporter, I did ask her, and she said that it took 100 phone calls / emails to get the data. And they estimated a similar thing, about 1,000. This is in mid-November. Now, so at that point, we thought, well, we don't have to do the study because clearly that something's going on. The government now should be aware and maybe are going to do something about it. But this is a statement made on December 13 after all this from the public health secretary, and they're saying there's no problem. And look in red what they're saying. They're saying in previous Decembers and Januaries we've seen these many deaths, so there's nothing to be concerned about. And that is, to anybody who knows anything about demography, you know that that's not that is not something you can do. The death rate in January and December are higher by quite a bit compared to other months because of mostly, I think, the flu or other seasonal diseases. So that was it was actually quite astonished that public health secretary would deny that something bad was happening. So when we saw this, we decided to keep going with the study. So basically to add more evidence to the existing evidence that something had happened. So the other thing that we saw is the New York Times did share the data with us. They sent us a PDF, and that was fun. Extracting the data from that was a lot of fun. It's like 100 lines of code. That is what the data looks like for 2017. I'm stopping right at the hurricane. This is daily data now, not monthly. And you can see very immediately that there's something happened after that, a big jump. But this is what the rest of the data look like. So what's that? Why is it going down like that? So one thing I learned was that, I think every demographer knows this, is that it takes a little bit of time to get all the deaths in the registry. This is totally expected that you would have a drop off like that. It takes time to get them into the system. So that's not, there's no hiding data here. That's just expected. But the point of that I'm making is that another question we had is it still happening? Are we still observing more deaths than expected? So we continue with the study. The general idea was to do we did a survey that was clustered because people were going to go to houses and ask questions. We divided the counties into how far away from a hospital they are. We wanted to have it represented. The areas in blue are places that are far away from a hospital, and they are tend to be in less populated places. So if you just do a regular survey, you won't get to them. So we found the neighborhoods we would go to. Those are the selected ones, and then we sent people there. So here's what there was like 50 students that would get a little like an iPad, and this is what would show up. The red is where you have to go. So they would go stand in that spot and then go to the closest house and ask. So the question was, has anybody died this year? What month? So then we have the data. This is what it looked like. We went to 10,000. We went to 3,000 houses. 10,000 people were included in the survey. And that's very little. You're going to see that in a moment. So this is the result. This is before the hurricane, the reported deaths from the survey and the age. So that gives you an idea what normal year looks like. And then this is after. We also asked them if they had problems accessing health services, and a lot of them answered that they did. So when you take this data and you now estimate the excess deaths from this, you get a pretty large number. We got a number about 4,000 was the point estimate. But I don't know how fast you can do the calculation in your head, but this is a very, very small sample for a rare event like death. So the confidence interval was quite big. It was something like, I think it was like from 800 to 8,000. Well, so I learned another lesson that I'm going to share with you about reporting things to the press. So we wrote the paper. It was published in May 29, 2018. And I was concerned about the that this message would be a little exaggerated. There's a little bit of you could see how this could be sensationalized. So I asked for the press office at Harvard to please send me the press release so I can edit it. And it was good that I did because it was not reporting the uncertainty in the estimate at all. So I made sure to tell them, you have to report confidence intervals or something in every single statement, because the last thing we want is to have the newspaper tomorrow say, Harvard professor says 4,600 people died in the Hurricane Maria. So that's exactly what happened. I think it was a journal. I didn't think of that. The journal also puts out a press release. So this is when I try to explain that it wasn't. No, see that's our PR office screwing up. So that you can see that you see what they said there. So I immediately corrected it. Mine, I mean, that's even reporting a confidence interval like that, I think is a mistake. Because what does that mean? I mean, we don't have the precision to put numbers like that there, right? It's like it should have been something like more than 800, maybe would have been a better statement. But it went viral and got all kind of press coverage. The number became like this symbol. So this is the mayor of San Juan. She made a hat, and she was in The Simpsons because Trump attacked her, and she became famous for that. We got Hillary Clinton saying it. And then actually, this is something that I thought was nice. This is they had an activity where people because people were really angry at the government at this point because they had been lying to them for a long time, and they had this activity where shoes of the victims were laid out, and they wanted to see if 4,600 would show up. I don't know if there's 4,000 there, but there's a lot. So it's very sad to see this. Well, getting back to statistics. The governor of Puerto Rico was grilled by Anderson Cooper, interestingly, because doing a survey like this is not what you do to estimate excess death in a place that keeps records of deaths. It just it's not what you do, because as you saw, it's hard to get precision unless you do a massive survey. But when we explained, we agreed with that, but we said, but the government didn't want to share data, so this is how we did it. It's another way of estimating. So that's interestingly enough, that's what Anderson Cooper was grilling him about. Like, why don't you share the data then if you're saying that's not right? And then he said, help to pay if they are not available. I want all governors to say that every day. So after this interview, right after this interview, then the data was made public. So now we have the data. So here's, this is 2017, 16 and 15 every day. And there's a little smoother going through. You can see it go up in the winter. This is 2017 before the hurricane, and here's the data after. And the reason I show you this is because anybody who had access to this data didn't, I mean, it's pretty obvious that something was going on. You don't have to necessarily even fit a model. That is clearly a visualization that clearly shows that there was a big problem in your health system. So once we got this, we wanted to get a more precise estimate. So we designed a model, and because we had daily data, which is not that common in these kinds of work, we thought we would do it for daily data so we can get that sharp beginning of the event. So we wrote, this is now published, like right now after all these years, got published in epidemiology, and it's an approach and a package and an R package that lets you estimate excess deaths in situations where you have daily data. So the daily data makes it a little bit more complicated. So some of the challenges I'll share is that one is that the demographics of Puerto Rico are very complicated. If you see at the end how the older bars grow, it's changing quite fast. So if you're going to estimate these things, you have to take that into account and know that if it's changing that fast, you might have some errors there too. The other thing is that right after the hurricane, a lot of people left. But that's not, the census doesn't capture that because the census does yearly surveys. So we connected with a cell phone analysis company that gave us data for cell phone. No, they didn't give us the actual cell phones. They just gave us the counts of Puerto Rican cell phones that were elsewhere by day. And you can see the big dip. This is our estimate based on the cell phone data. And you can see the big dip right after the hurricane. So that, when you're estimating excess deaths, you have to take that into account. There's less people, the denominator of the rate is smaller, so it all goes up. So with all that said, this is what our software produces, an estimate of increasing death rate with confidence intervals. And it uses a smoother so it can, that permits jumps on the day of the event to capture spikes like the one we saw in Maria. And when we got the data, when Puerto Rico sent us the data this time, they sent us all the data from the beginning of times. And we actually saw a similar, we looked at the previous hurricane and we saw a similar little jump. That was also interesting finding. It's not as bad, but it is not great either. It's like 1,000 extra deaths. So that statistical methodology, when COVID came, we developed it for this, but when COVID came, we started getting requests for it. And so we decided to actually work on it and make it more rigorous and all that. So now that was a motivation for that. That's why it came out now later. So this is, the New York Times used it to estimate excess deaths in the US. This is back in May 13, 2020. I got a call from this guy, Nicholas Christoph, and he wanted to do this. And I helped him, like, yeah, sure, just tell me where to get the data. And it was pretty easy to do because I already had the package from working on it on Puerto Rico. And then this group in Europe had like all data from all the European countries, and we helped them use it as well. Okay, so I'll end there. I acknowledge a bunch of people that were involved in all these different projects. Thank you. Any questions? Okay, and then from the Zoom. Oh, okay. Got it. All right, throw on this thing, no? Questions? No questions? Okay.
You talked about these sort of three different projects. I'm curious what new technologies you're excited about now.
Sure. So the question was, what technologies am I excited about? Oh, right now, the main one we're working on that's very new is spatial transcriptomics. That was the other topic that I suggested, but that lost the poll. And what it does is it measures. We have a collaborator that has a version of it that almost can do single cell level gene expression counts, also telling you where on the tissue slide it is. So the reason that can have a lot of applications, especially in cancer, is because we can see, for example, if certain immune cells are somehow different, if they're near the cancer and not, and we can compare cases where they in this immunotherapy techniques, that if there's something we can look at that could predict when it's going to work and when it's not. This thing has a lot of applications. So that's the main one probably right now. It's always changing. I should say, like every year there's something very cool coming out that's, has a lot of challenges that statisticians can help with. No, throw it. Ah, there you go.
final estimate for the excess mortality in Puerto Rico?
Good question. So it's about 3,000.
Okay, so not too far off, yeah.
Yeah.
But
Well, okay, 3,000 was because if you look at the plot, let me make it big. Well, it's a little bit more off than it sounds because that went. Oh, that's the other one. Look at where it ends. It ends in like April. Our estimate of 4,000 was for December.
okay.
So I think, but I think we were that. It was probably around 2,000 something in December. We can. I have that calculation somewhere, but it was in the confidence interval.
Yep.
Yeah.
Hi, great talk. I see that all of your applications are pretty different that you talked about today. So was a lot of the statistics that you were working on these different projects kind of similar, or like you kind of learned a new different skill set every time you picked up these different projects?
Yeah. So the question was, were the projects were applications were very different? Were the methods different? And yeah, they were. I don't. In general, my approach to apply statistics is ask what the problem is, look at the data, and then figure out what the best method is. So if you do that, you'll undoubtedly end up using very different things.
Okay, I have one more question. And then I kind of see that there's still like excess death, like even after the hurricane hit. So is that kind of due to like people not being able to get access to healthcare and just other like downstream effects from Hurricane Maria? Because you did have some points there from like indirectly death from Hurricane Maria. So was the government also trying to, because they were like kind of saying directly death from Maria. So I don't know how they were counting those directly and indirectly as well.
Yes. So great question. So the question was how many of these were these deaths due to the hurricane or some indirect effect? I'm summarizing for the Zoom people. It's mostly indirect, and it's a little detail I didn't mention is that electricity was out for about 90% of the population for 4 months. So that means if you have insulin dependence, the fridge might not be working. If you have a respirator that you need, it doesn't necessarily work. And I also think the stress, this is now speculation, the stress of being without electricity for 4 months, it can also have an effect on especially the elderly. That is something we did it by age, and it was definitely the majority of the excess deaths came in the older groups, not surprisingly, given that they're indirect effects. I know. I'm really eager to see it happen.
I was waiting to catch it.
Yeah.
Yeah, so fascinating. So you're sort of known for looking at the data and looking at it hard and very critically and being very skeptical and in some sense of a signal among the noise. So do you ever get situations in these data sets where there's you just don't believe data points? This one has to be wrong, and how often do you throw it out? I mean, how do you deal with that sort of setting, which you must come across all the time?
Yeah, that's a difficult one. I think about I think so the question was, how do I know when I must encounter situations? I definitely do, where it looks like the data is just not right. And how do I deal with that? It's a case by case. It's it's something I think about, like, how can I teach this to others? I mean, people I'm training, because it's such an important thing. I see a lot of times trainees come to me and show me something and it's obviously wrong. I see it pretty quickly. How do I get them to see it too? Like I point it out and I explain, but I want them to learn to see it before, not even come to me with it. So the only answer I can give to that is that first, understanding this is why statistical theory is still important. If you understand statistical theory, like for example, the as an example, that the average of correlated data has a higher standard error than independent data, just to throw out an example. Knowing things like that helps you detect and figure out why something is not making sense. So just having a general understanding of statistics, theoretical statistics, helps a lot. Another one is plotting. Making a lot of plots just reveals things that just don't make sense. Being super skeptical about things that are unexpected and trying to find holes in it. I mean, these are just the things that I can answer right now, but it is a hard thing to teach and to describe how it is that we learn to do it.
And then
You know, I
throw
tell you
out
one, I'll give you
the
one
data?
example. A recent example was that we were finding now I'm helping the government of Puerto Rico analyze their vaccine effectiveness data. And we were seeing very, very different effects between age groups. And it was assembly. I mean, it just didn't make any sense. And then eventually you figured out it was that the population estimates were wrong. So the unvaccinated, as you get closer to having everybody vaccinated, that denominator gets smaller and more susceptible to small errors. So we used, as soon as we switched to another of the census estimates, it all went away. But if we just use the official data, we would have gotten wrong answers. Wait. All right,
I
ready?
wanna do it.
Ready? I don't know. Watch out.
All right.
Touchdown.
All right. I don't have my phone. So I have a question. It's relatively about switching between these projects. Do you have a period of time you feel you have stopped doing microdata and then trying to do new things? Or like, do you feel that transition of technology has significantly impacted the proportion of time spending on your old projects, new projects?
Yeah, so this is a time management question. So how do I decide when to switch projects? So the microarray one would just that, because in genomics, I mostly follow what my collaborators are doing. They ask me questions. That one just happened because my collaborators start coming to me with RNA-seq instead. So at some point, I noticed that nobody was using microarrays anymore. So my time got sucked into other. And another thing that happened there was that we moved to methylation because my collaborators that I was working with, that's what they were interested in. So that was another transition. So I just had those are the projects that were most interesting to me. So I just started working on that and stopped working on the older stuff. So in my case, it's mainly driven by what my collaborators are doing.
Sounds good. And also another question is about the cause of death. So I do work a little bit on verbal autopsy, which is trying to interview relatives of people who just died and asking them what are the symptoms that they observed in the period leading up to the death. And one common question is that how can you be sure that it's not because the hurricane is harvesting people who are already very frail, or
Yeah.
do you have any misclassifications of cause of death, and how did you deal with that?
We do. We have cause of death as recorded in the demographic registry. We see the question was was this a harvesting effect where a lot of the people who were going to die anyway just died earlier? So first, let me answer why I don't think that's the case here, and it's because we don't see the curve doesn't go below 0. So, right, so that's what the harvesting effect would look like, and we actually see that in Puerto Rico data for the chikungunya outbreak. There you see the harvesting effect because it went up, but then it came under. So but here we don't see that. But the other way we could try to answer that is by looking at the reasons, and there was basically a lot of heart attacks and respiratory disease, but actually that could definitely still be harvesting effect, right? So The only answer I have is that it was that the fact that we don't see, we don't see a dip later. But also it could be a little bit of harvesting. Yeah.
So you still count those harvesting
Yeah,
towards.
so I guess I would say that what we're doing is we're just for this particular period of time, we observe these many excess deaths. Yeah, I would try to, when I'm making statements to the press in particular, I try to stay away from causal statements like that. Like, we know the hurricane caused it. I just say, look, this state to this state is what we see.
Sounds like you don't want another CNN interview.
Okay. Well, thank you again.